Hi, I'm Elaine Vickers and welcome to my video series on cancer treatments. In this video, I'm going to describe a group of small molecule treatments called kinase inhibitors. There are many examples of kinase inhibitors used as cancer treatments, drugs like imatinib, osimertinib, sorafenib, and lots of others. But what exactly do kinases do? Kinases are enzymes that make stuff happen inside our cells. They take a protein that isn't doing very much and they switch it on so it becomes active and it starts doing its job. They do this by first grabbing hold of a little chemical compound called ATP, adenosine triphosphate. It's called this because it has 3 phosphates, triphosphate. The kinase takes one of these phosphates and they transfer it onto a target protein. A phosphate is a phosphorus atom surrounded by oxygen atoms, and it's packed with energy. So when a kinase attaches a phosphate to something, that can be enough to bring it into life and swing it into action. The activated protein might now be able to interact with other proteins, or it might attach to DNA and switch on a gene, or maybe it's a kinase enzyme itself, and now it's able to phosphorylate something else. In other words, kinases attach phosphate to things and that makes stuff happen inside cells. By the way, if you like my teaching style and if you want some education for your team, then do get in touch and there are links in the description below. So what do kinase inhibitors do? Not surprisingly, they block kinases. Most kinase inhibitors are small molecules that slot into the little ATP binding pocket of a kinase enzyme. If ATP can't bind, then the kinase can't get hold of a phosphate and it can't activate its target. This is especially useful in cancer cells, which often contain faulty kinases. These kinases force the cancer cells to grow and multiply and prevent them from dying, even though they contain lots of DNA damage. Because the faulty kinase is driving the behavior of the cancer cell, a kinase inhibitor can cause cancer cells to die. So what about TKIs? This abbreviation is often used for kinase inhibitors, but you've probably noticed that there's a T in there which doesn't exist in kinase inhibitor. That T stands for tyrosine. To explain that, I need to go back a step. You might remember that all proteins are made from tiny building blocks called amino acids. If I've already lost you, now might be the time to watch a couple of the Cancer Basics videos about genes and proteins. Each protein contains hundreds or even thousands of amino acids. There's about 20 different ones and they're linked together in a specific order in each protein. Kinases are particular about which amino acids they attach phosphates to. A tyrosine kinase is a kinase that attaches phosphates only to tyrosine amino acids in its target proteins. So a TKI, a tyrosine kinase inhibitor, is a drug that blocks a tyrosine kinase. Now personally, I don't like the name TKI. One problem I have with it is that as soon as you use it, your listener thinks, well, what does that stand for? And if you tell them, they probably won't understand your answer and they're no better off. A second problem I have with it is that the term often gets misused and used to describe treatments that aren't TKIs. Some cancer drugs really do block tyrosine kinases and you can call them TKIs. This includes drugs that block EGFR and HER2, MET, RET, ALK or BTK. All these proteins are tyrosine kinases and the drugs that block them are bona fide TKIs. But I've lost track of how many times I've seen or heard BRAF inhibitors, CDK inhibitors or mTOR inhibitors referred to as TKIs. And although these proteins are kinases, they attach phosphates to serine and threonine amino acids and not to tyrosines. Even more confusingly, drugs that block PI3 kinase enzymes are also called TKIs, even though PI3 kinase doesn't phosphorylate a protein. It doesn't attach phosphates to any amino acid. It attaches phosphates to a tiny lipid molecule in the cell membrane. Then we have the drugs that block enzymes that aren't kinases at all, such as PARP and KRAS, IDH1 and IDH2 or the HDAC enzymes. I've even seen the term TKI used to refer to drugs that block proteins that aren't even enzymes, proteins like smoothened or BCL2 or menin, yet the TKI label still gets used. I've split the summary of this video into two, a simple version and a more sciencey one. We'll start simple. Kinases are powerful enzymes. Cancer cells often make faulty overactive kinase enzymes because of mutations in their DNA. These faulty kinase enzymes drive the cancer cell's behavior and keep them alive. Kinase inhibitors are drugs that block kinases, and they can be used to kill cancer cells that rely on a kinase for their survival. The more sciencey summary is that kinases are enzymes that attach phosphates to things, and that makes the thing active and it starts doing its job, and most kinases attach phosphates to other proteins, and often those other proteins are themselves kinase enzymes. Kinase enzymes use ATP as their source of phosphate, and kinase inhibitors stop this, often by mimicking ATP and occupying the ATP binding pocket of a kinase enzyme. Some kinase inhibitors are genuine TKIs because they block a tyrosine kinase, but many of the drugs that get called TKIs really shouldn't be. And don't forget, if you like my teaching style and you want some education for your team, find me on LinkedIn or contact me through my website. There are links in the description below.